Good afternoon, everybody. Today I am here to talk to you about the physiological aspects and effects of glaucoma on the eye and how the disease affects vision through the optic nerve and the nervous system. I'll also be discussing recent developments in potential advancement for potential treatment of the disease. So what is physiology? So physiology is the study of how molecules, cells, tissues and organs function in health and disease. And to understand glaucoma, we must first understand the optic nerve and the physiology of the eye. So the physiology of the eye. So the eyes are a pair of spherical organs located within the orbits of our skull. The eye is one of the most complex and important sensory organs in the human body. It provides the ability to see both bright and dim light and has the ability to focus on objects both close and far. The eye has three types of cone cells, which are able to distinguish millions of distinct colours and produce high-quality images that the body relies on for many daily activities. So here we've got an image of the eye. So you've got the cornea at the front, and that's just here, this part. The pupil, which is the hole that controls the amount of light entered through the eye, which is this part here. Then you've got the iris, which is the coloured part of your eye, which is up here. You've got the lens, which focuses the light onto the retina, which is in here. Then you've got the sclera, which is the eye's strong outer coat, which is this section down here. Then at the top you've got the retina here, which receives the light and records visual messages. And then at the back over here, you've got the optic nerve, which carries the visual messages from the retina to the brain, which is the part of the eye that gets destroyed in glaucoma. So the optic nerve connects the eye to the brain, and the optic nerve is responsible for carrying impulses formed by the retina. So the retina is a thin layer of tissue that lines the back of your eye, and it's located near the optic nerve and serves the purpose of sensing light that has been focused by the lens in your eye and converts this light into neural signals, which are then sent to the brain for visual recognition. The retina is responsible for creating impulses, and these impulses are dispatched through the optic nerve to the brain, which then interprets them as images. And glaucoma is a name given to a group of eye diseases where your vision is lost due to damage to that optic nerve at the back. So according to Glaucoma Australia, there are currently 300,000 Australians with glaucoma, and generally there are no symptoms or warning signs present in the patients. And the patients that suffer from the early stages of this eye condition have a loss of sight, and it's usually slow and degenerative over time. And usually the disease causes a considerable amount of loss of their peripheral or side vision before awareness of the problem is realised, so they have the disease and they're unaware that they have it. I've discussed the physiology of the eye, but to understand glaucoma, you need to understand how the disease works itself. So firstly, the primary problem in glaucoma is damage to your optic nerve, but this happens when intraocular pressure is too high, or fluid pressure in your eye builds up and causes damage to your optic nerve. So the degree of damage to the optic nerve varies between person to person. And in glaucoma, the pressure builds up from unhealthy changes in the drainage tracts in the front of the eye that can come with aging, like reduced flexibility and increased scarring of that tissue. Some individuals with high levels of pressure don't always develop nerve damage, and others with normal pressure levels may develop nerve damage. So, according to Glaucoma Research Foundation in Australia, glaucoma is the second leading cause of blindness in Australia, and the vision that's lost through the disease cannot be regained. The disease is usually more apparent in older individuals, but there are cases where children are born with it or young adults also develop the disease. So researchers seek new approaches to better protect the eye from glaucoma, and under increased pressure of glaucoma, the scientists want to help the neurons in our eyes better protect themselves. They're working to see whether agents that activate the innate neuron productor sigma-1 receptor, or S1R, can do this. And current glaucoma therapies work to reduce pressure in the eyes by increasing the outflow or reducing fluid production. Dr. Catherine Bollinger, an ophthalmologist and glaucoma specialist, has evidence that a good additional strategy would be directly enhancing the production of neurons in our eyes, called retinal ganglion cells, by directly activating S1R. So we may be able to protect the retinal ganglion cells more long-term, she says. And she's been given a $1.5 million grant from the National Eye Institute to research this. Her research looks at how S1R stimulation has been shown to protect retinal ganglion cells in other disease models, as well as glaucoma, but hers is the first work in actual glaucoma. Her work over the past year has focused on the impact of activating these S1R in the astrocytes. And a new study looks at whether activating S1R with existing drugs already used in patients for other reasons also enables neurons to better protect themselves. The high pressure glaucoma produces inside the eye seems to transform the astrocytes, the type of brain cell that provides nutrition and other support to the neurons, into a nemesis that instead releases toxically high levels of things like nitric oxide and reactive oxygen species. In a rat model of glaucoma, Bollinger's team is further exploring the impact of activating S1R with known antagonists like the pain reliever pentazoacine and commonly used antidepressants called selective serotonin reuptake inhibitors. They're looking at their impact on neuronal survival and function at the level of support from protective proteins like brain-derived neurotrophic factors, which aids neuron survival as well as new neuron growth. One question Bollinger and her team want to answer is how can these drugs protect or rescue neurons in the face of glaucoma? And they're looking again in culture in the animal model and what they suspect S1R's role in mediating the reactivity of astrocytes. Astrocytes' increased reactivity in turn appears to prompt release of neurotoxic substances like the high level of nitric oxide, which further raises pressure and reactivity. But when scientists treat the usual supportive cells with S1R agonists in a dish, the cells back off their bad behaviour and appear more willing to release good things like brain-derived neurotrophic factor. The study shows that some astrocyte changes in response to pressure may actually be helpful to neurons, while others are deleterious, which is something she wants to understand. As we kind of piece together specific consequences of astrocyte activation, we might be able to better manipulate astrocytes so they will just protect neurons instead of damaging them, she says. And there's good evidence that changes to astrocyte behaviour occur rather quickly in response to increased pressure in the tight confines of your eyeball. She states while patients with glaucoma may experience some level of decreased vision, a minority will experience blindness, and there's no way to predict who those patients are and no additional means to intervene. Given our ageing population and the fact that glaucoma is prevalent, we do see blindness secondary to glaucoma. Bollinger says, conversely, some patients may never experience any vision loss, for the studies of the S1R agonists are injected into the abdominal cavity, but a topical application could be developed if findings continue to hold in the future.